Okay.
Good afternoon to one and all. And now we are moving on to the third session of today's invited talks and lecture series. Today we have the eminent personality, Dr. Ruby John Anto from RGCB Trivandrum on the topic Interdisciplinary Research, the Future of Cancer Care. This session will be chaired by Dr. Anto PV, Department of Commerce. I request Dr. Anto PV to invite and chair this session.
Good afternoon, everyone. On this auspicious event, I want to call upon Dr. Ruby John Anto for this session. She is presently working as a scientist G at Rajiv Gandhi Center for Biotechnology, Trivandrum, and renowned for her work in cancer research. She has earned her PhD from Amala Cancer Research Institute in the field of biochemistry. To showcase her academic excellence, she has been awarded the International Global Health Award from Brazil in 2019, along with the Excellence Award for Outstanding Performance from RGCB in 2015, combined by the National Women Scientist Award by DBT in 2004. She has been awarded twice with a fellowship from the National Academy of Science. She has also received 4 patents at the international level and 2 are pending. She has successfully completed 15 significant major projects and has published 90 scientific papers, out of which 10 have high impact factor with 6,000 citations. She served as reviewer of many journals. I once again invite Dr. Ruby ma'am to take this session. Please, ma'am.
Thank you for such a nice introduction, and I thank the organizers.
Unmute.
Unmute. Hello? Hello?
Yes, ma'am. Okay.
Unmute എന്ന് പറയുന്നു.
Hello? No issues. You can continue, ma'am. It's audible here.
Okay. So I thank the organizers of this conference, especially Dr. Anne-Marie, for inviting me to give a talk here. And coming to the interdisciplinary research and the future of cancer care. So,
Are you facing any issues?
So the treatment of cancer, of course, it requires a lot, the expertise of lots of the people from different fields. So in interdisciplinary research, it is a teamwork of different individuals which integrates the information of the research or whatever the efficacy or the work of different teams, and we integrate the information, data, techniques, tools, concept of and theories of all these teams, especially to understand a specialized knowledge to advance the fundamental understanding or to solve the problems. That means several people are uniting for a single cause. That is interdisciplinary research. So here we will see the application of this interdisciplinary research in cancer care. So for a patient, a cancer patient, I mean, so see the different people from different fields are uniting to treat the person. Okay. So it's a big team. comprising of it's not just a doctor patient relation. So many peoples have to unite together, okay, medical or non-medical, for the cancer care. So then regarding the remedies for cancer, actually the measures, the first thing to do is better, the most effective thing is prevention. Okay, so during the, we all say that it is better to prevent than to cure. So here also it is very much applicable. So how to prevent cancer? The first one is chemo prevention. That means there are certain compounds, non-toxic compounds, which can prevent the occurrence of cancer. And the other one is change in lifestyle. 80% of cancer is occurring due to environmental factors. Okay, so if we change the exposure, that means tobacco, alcohol, and exposure to chemicals, so many things. Okay, smoke, the area in which there is heavy pollution, all these things can cause cancer. So if you change your lifestyle, and obesity, that is also another important factor for contributing to breast cancer, hormone related cancers. So if you change the lifestyle and stop exposure, then also we can prevent the occurrence of cancer. Next one is, if all the cancer has already occurred, it comes to therapy. So there are different modes to treat the cancer. The most important treatment treatment options are surgery, chemotherapy, and radiation therapy. And chemotherapy means the use of chemicals to treat cancer. So the chemo prevention, coming to chemo prevention, the process of carcinogenesis is a multi-step process. Okay, it just does not occur just by a single step. Okay, the first step is initiation. That means the mutation of the normals to a normal cell to mutated cells. This is an irreversible process, but just because the cell is initiated, it doesn't become cancer. This mutated cell have to proliferate to a big mass to become a tumor. So there are compounds which can stop this initiation step. This is called primary prevention. Okay, so you can stop the carcinogenesis process in the first step itself, that is stopping the initiation step, that is the primary prevention. Second is this cell to divide and to become a huge mass. That process is called promotion. For example, this in the case of tobacco is a initiator. Okay, cigarette smoke, all these things are initiators. But in the case of alcohol, it cannot initiate a cancer. It can only promote cancer. It is a promoter, alcohol. Okay, so this step also can be blocked using chemo preventives. They are called secondary preventives. Okay, and still at this stage also, if we remove this tumor, we can stop the development of a metastatic cancer. So from this step, it will progress to a benign tumor and malignant tumor, which will invade the whole body, different organs of the body. This step is called progression. Okay, so when you block it in the promotion step, it is a secondary preventive. And when you stop it in the this benign to this malignant state, it is a tertiary prevention. So there are different compounds, natural compounds, which can stop the carcinogenesis process at different steps. Okay, so this is how chemo prevention works. So these are different, so many plants are there and the ingredients of these plants are known to block the process of carcinogenesis, either initiation step or promotion step or progression step. Now coming to chemotherapy, I told it is the treatment of cancer using chemicals. So depending upon their mechanism of action, they have been classified into different groups. So there is no compound or chemotherapeutic drug which can identify the cancer cell, okay, and kill only the cancer cell. It is not there. There is no such compound. So the compounds are targeting cells which divide very fast. That is the principle of selecting a chemotherapeutic drug. It will kill the rapidly dividing cell. Okay. So when you kill the rapidly dividing cell, there are cells within the body which also, normal cells, I mean, they also will divide very fast. That is the cells of the hair follicles, cells of our digestive tract, all these also divide very fast. So when you take a chemotherapy, these normal cells also die. That is the reason for the heavy side effects and making the person very sick. They cannot eat food because the digestive tract is completely gone. Even drink water, that is also very difficult. So some people, they don't eat food and die because of these side effects. And another problem with chemotherapy is if a drug is not that toxic, then it will be very costly. Okay, that is another thing. So if you cannot afford the cost, then you have to go for toxic chemotherapeutics. There are different choices. Okay, so that is another problem with this chemotherapy. So when we reduce the dose, okay, if you think that, okay, I will reduce the dose so that I won't have the side effect, I don't have to take that much money also, then what happens is some of the cells won't die. Okay, so there will be some surviving cells. Okay, this is called the cancer stem cell like population. Okay, so this tumor, okay, this cell will, okay, it will be resistant to the current drug and it will multiply. Okay, so then they will expand and will become a colony so that you cannot, it will be resistant to the chemotherapy. Okay, so this is the problem with the low dose of chemotherapy. So we should take, when you are going for a chemotherapy, it should be the apt dose. Otherwise, you will get chemo resistance. So it is in this scenario, the concept of chemosensitization emerged. There are certain compounds which, while not very active alone, okay, they may not be able to kill cancer cells alone, okay, but they have the ability to heighten or increase the efficacy of the chemotherapeutic drug, the conventional chemotherapeutic drug. That means all the chemotherapeutic drugs, the conventional drugs, they at the low dose, that is the reason, one of the reason for this chemo resistance. The low dose of all conventional chemotherapeutic drugs have the efficacy to induce the survival signals. Survival signals means the proliferative signals of a cancer cell. So if you are giving a cancer drug, chemotherapeutic drug, it has the property to induce the survival signals at low doses. That means the cell become resistant. So at this stage, these drugs, these chemosensitizers, that means the compounds which can downregulate the survival signals induced by the conventional drugs. Okay, so these are the compounds, curcumin, resveratrol. It is found in the skin of black grapes, EGCG from green tea, gingerol from ginger, green tea also. Okay, so there are several compounds which can chemosensitize by downregulating the upregulated survival signals induced by these chemotherapeutic drugs. So they act as a combination. Actually, the killer is the chemotherapeutic drug itself, but this chemosensitizes when we pre-treat the cancer cells before treating with the conventional drug with this type of compounds. These compounds will be non-toxic, so it doesn't induce the toxicity. So and it is not very costly also, so the cost also will be less. So you can reduce the cost of the treatment and the toxicity induced by the conventional drug by using these type of chemosensitizers in combination with the conventional drugs. This is the process of chemosensitization. So our lab is focusing on identifying, using our Kerala biodiversity is a treasure house. So many plants, medicinal plants having different compounds. Okay, so we are trying to identify the active component which can prevent or treat or sensitize cancer cells. Okay, chemotherapy means treating, and chemopreventive means block the carcinogenesis process, and chemosensitize the conventional drugs, and to identify the mechanism of action of these drugs. That is the focus of our lab. So what we are doing is we shade dry the plant, and because the several compounds will be sensitive to light, so we won't keep it in sun, so we keep it in shade and dry it, then powder it, then we use a polarity graded extraction. Different extracts will be produced, made by shaking in orbital shaker. Then you evaporate off all the solvents and take the extract, and we just put it in cancer cells of different origins. Okay, so when you treat the cells with if this is cancer cell, we keep a control, and when you treat the cells with this our drug, if the cells are dying, okay, if there is a decrease in the number of cells, that means the compound has a cytotoxic activity, but it doesn't mean that it is a drug. Okay, it is not even a compound, it is just an extract. Okay, so we can conclude that that extract has some cytotoxic cell killing activity. So what we do is the same extract we put it. If we are getting an activity in breast cancer cells, we put the same extract in normal breast cells. Okay, immortalized cells we put. Then if you are getting the same cytotoxicity there also, it is not a drug. Okay, you don't need to further proceed. But if you are getting the same cytotoxicity at 5 times the dose, then there is a probability of evaluating it further. Okay, if you put potassium cyanide also, all the cells will die. It doesn't mean that it is a drug. So we have to compare the cancer versus normal cells. Okay, after getting this, after the preliminary screening, we do the separation of this extract using different methods. There are so many methods for purification. Okay, column chromatography, so many things. Okay, thin layer chromatography, so many things are there. And we separate out the single compound. And we do the in vivo. You can do so many in vitro assays also using this isolated compound. Then to find the, maybe if a compound is cytotoxic in vitro culture, it may not be active in vivo condition. I will come to that. So after that, we will go for testing in vivo means within the body. In vitro means within the cell culture system. So we inject the cancer cells in immunocompromised mice. If you inject a human cancer cell into normal mice, it will not grow because of the immune rejection. So we use immunocompromised mice and induce cancer cells. And after the cancer start developing, that means after 15 days you can find a small tumor, then you start treatment. Okay. So these are the different modes of drug administration through the parenteral way, that means through the subcutaneous, and oral and tail vein. You you can inject just like IV. Okay. So for example, if you keep two animals, this will be here. We won't give drug. So here we will be giving drug. Okay. So here the tumor won't be developed, or there will be reduction in tumor size. Okay. So that it is a mode to evaluate the efficacy of drug in the in vivo system. Yeah, you can see actually here we did not see any tumor from outside, but when we surgically removed, we could get a tumor. Okay. Then the next stage is the clinical trial. For that, the first we will be doing a human safety trial. Then it will be done in patients, and there will be phase 1, phase 2, phase 3 trials. Then it is going to market. That is the drug delivery. It is in the nutshell of the drug discovery program. It's not very easy. As I said, it will take years to make for a drug to come into the market. So these are the compounds which, these are the molecules and extracts or whatever is I studied in our lab. The most studied compound in our lab is curcumin, the color pigment of turmeric. We have studied it as a chemosensitizer and as a chemopreventive. And these are the other compounds. And this is our star compound. Okay, utroside B. This is a compound identified in our lab from the Solana diagram, and it has been patented and commercially transferred. I will come to that. So we found that curcumin can sensitize cervical cancer cells to paclitaxel. Paclitaxel is a very costly drug. It is isolated from the skin of a tree called Pacific yew. It will take 100 years to for a Pacific yew tree to grow to its full size, and at least 3 trees are needed to treat one patient. So you can imagine the cost of the drug. And when you use curcumin along with this, I mean pre-treat curcumin, we can reduce the tumor size. We can reduce the dose. Half the dose of paclitaxel will bring more than the effect of the this is paclitaxel alone. This is the combination, and we have patented this combination. And this the students who did this is Dr. Smita. She's working in Calicut University currently. This is Dr. Shrikant. He is a postdoc in US currently. So this is another model for cervical cancer. This is animal cervical carcinogenesis model. It is a chemical induced model. Okay, here also we could find a very good tumor reduction for the combination. Okay. So the problem with curcumin, if you put curcumin 25 micromolar in any cancer cell in culture, in vitro, okay, in cell culture, any cancer cell will die. Okay. But the problem is the compound is not soluble in water because we are adding it in DMSO. In cell culture, we are adding in DMSO. But DMSO is a toxic chemical and you cannot give it into the body of the animal. Okay, so you cannot give curcumin dissolved in DMSO to the animals or human. So the problem is if you give orally, it is a non-toxic compound. You can give any amount. Even studies have reported even 9 grams of curcumin per day is non-toxic to the body, human trials. But the problem is after giving this much curcumin to the person, and if you take the blood of the that person, the serum concentration of curcumin will be 5 micromolar, less than. Okay, around 5 micromolar. That means you cannot achieve the concentration to 25 micromolar of curcumin within the body. Okay, so that is the problem. I told you it is that's why we thought of using curcumin as a chemosensitizer. Otherwise, we could have treated it as a chemotherapeutic. We could have considered, but it is impossible to bring that concentration in the in vivo condition. And another problem is it won't reach the different organs of the body, poor bioavailability, and the tissue retention is very low. After some time, if you see the body, nothing will be there. It will be completely driven out of the body within 3 hours. Okay, so these are the drawbacks of using curcumin. There are so many compounds which show very good activity in the in vitro system, but in the in vivo condition, they won't be effective. Okay, so in this condition, we thought of using nanoparticle mediated drug delivery system. And this is this application of biodegradable polymeric particles in the scale of nanometers. And they can use a controlled release dosage. Slowly they can release the drug. And we studied we screened several nano formulations loaded with the curcumin. Curcumin loaded PLGA, here polylactic and glycolic acid, that is the combination PLGA, and folic acid PLGA. It is conjugated with the folic acid, and this is acrylic acid. So many molecules. And we have also used the curcumin adsorbed nanogels also, PA nanogels also. All this we tested in the in vitro condition. First, we did a screening, which one is to take for the in vivo conditions. So this is PLGA. It can increase the solubility of the. It's not solubility; it becomes water dispersible. Aqueous. You can see here. This is curcumin. When you put curcumin in water, aqueous media, it will completely suspend. But when you make a nano formulation, it can be dispersed. We are here. This is the first interdisciplinary collaboration. Okay, here Dr. G.S. Vinod Kumar from our chemical biology department, he has made the different nano formulations for us. So this is one interdisciplinary collaboration. You can see this is curcumin, pure curcumin suspended in water. You can see the whole curcumin in the bottom. It's not soluble. This is curcumin dissolved in DMSO. This is completely soluble, but the problem is it cannot be used in vivo. So these are two types of curcumin he made. PLGA curcumins. These are dispersed in water. This is 75 to 25. That means that it is the ratio. This lactic glycolic ratio. One is 75 to 25. The other is 50 to 50. Okay. So both are soluble. I mean dispersible. Completely dispersible. We don't find anything in the bottom. Okay. So this was done by this biological evaluation of this work was done by my student, Dr. Arun Kumar. He also left the lab. So these are the different TEM images of curcumin and trapped PLGA nanoparticles. So we just compared the uptake, cellular uptake. This is nano curcumin 75-25, that formulation. Okay, so we can see the curcumin. Okay, fluorescence of curcumin green. This is 50-50. Here also we can see. This is curcumin in DMSO. Here we see. So it is showing that when you do that the nano formulation, we get the same effect of curcumin dissolved in DMSO. So this is in water. This is in DMSO. This only can be given to animals or human beings. This is curcumin medium. We don't see any fluorescence. That means it is not there. It is not attached. Okay. See, these are the blank form polymers just for as a control. And we checked the cytotoxicity. We compared the cytotoxicity of all these formulations in cancer, different cancers. So we found that almost all are same. We did not find much variation. We compared with the curcumin. So curcumin in DMSO. So again, people may think that what is the need of this nano formulation? It is curcumin in DMSO also is showing the same cytotoxicity, but we should remember curcumin in DMSO cannot be used as a drug. That's why this is in water we have dissolved, the other is curcumin in DMSO. And when we are getting the same effect, we can use the nano formulation in water. So we used, we produced 2 conjugates. Okay, we know the folic acid. Folic acid will be overexpressed in the surface of the cancer cells, cervical cancer or breast cancer, etc. There will be a strong expression of folic acid. So if you can conjugate our nanoparticles with folic acid, okay. So it will go and find the folic acid receptor will be on the tumor. So this folic acid will go and bind to the receptor. Then our drug also can bind there. So we made the folic acid conjugated PAA and PLGA nanocurcumins. We found both the compounds, both the formulations have very good. You can see here. Okay, this is blank. We don't see any curcumin here. Definitely it is a control. And curcumin DMSO. Here we can see, but again we cannot use it in vivo condition. This is just to compare. We are getting better efficacy than DMSO curcumin in the folate conjugated. Very nice accumulation of curcumin in the conjugated cells. And we compared using different clonogenic assays. So many experiments we did to compare whether it is okay or it is comparable. We can see that both are almost same. We got the same result, almost the same effect in both the formulations. Then we selected PLGA for our PLGA folate combination for our further studies, because this PLGA is a fast degradable and FDA approved drug, their formulation to a drug carrier as a drug delivery, because it will degrade very fast and the end products will be carbon dioxide and water. So it is not at all toxic. Here also PA is a biocompatible, but it will stay within the body. The degradation is practically very slow. Okay, and so here it is FDA approved for as a drug carrier for IV, IM, any type of injection. PLGA is approved, while PA is approved as a drug carrier for implants, to make implants. It will stay there. It is not toxic, but it will stay in our body. So that's why for our study we found the in vitro efficacy was same. We did not find any difference for both. Okay. Here also we can see we did not find much difference. So for our study, we selected the folate conjugated PLGA nanoparticles. So you can see the in tissue. This is the PP. We call that folate conjugated PLGA particle as PPF curcumin. Okay. So it is. You can see in 3 hours. This is the liposomal curcumin. Before using this nano formulation, we are using liposomal curcumin. See the difference in the intensity of curcumin in the tissues. Here it is very less. And after at 6 hours, nothing is there. It's completely gone in the case of liposomal curcumin, but still it is here. And again, 12 hours, still the fluorescence is there for PPF curcumin, but nothing is there for the liposomal curcumin. And 24 hours also, there is slight intensity retention of curcumin, of nanocurcumin, PPF curcumin in the tissue. So it will, so this is the graph showing the variation. So it is very clear that the nano encapsulation of curcumin will increase its tissue retention, the time. So this is just to show that folic acid conjugation, why we used the folic acid conjugate. This is just a confocal microscopic image showing the folate receptor on the surface of the HeLa. HeLa is a cervical cancer cell. So you see, okay. And when you uptake the curcumin, PPF curcumin, you see the curcumin on all the cells. So strongly it is bound. And this is another cell. This is Hackett cell. It is a normal immortalized cell. We don't find folate receptor here. And when you put the PPF curcumin, it is not binding here. Okay. So it gives it's a sort of site specific. The drug delivery is site specific. It will not bind to the normal cells, which do not have that much folate receptor, while it will bind to cells, any cancer cell, or if there is any normal cell, then also it will go and bind. It will go to any cell which has a very good folate receptor expression. And in the normal cell, we did not get a sensitization with the PPF curcumin in Hackett cells. So to confirm that, we again just incubated. This is the folic acid pre-treated. This is PPF curcumin treated cells. Then we first covered the cells pre-treated with folic acid. Then we added PPF curcumin. It is not seen. Okay. So that means when you add folic acid, this folic acid will already go and bind to the folate receptors. Then our curcumin folate cannot go and bind. So that means it is just to confirm that this is the mechanism. Okay. So this is the results of our in vivo study in cervical xenografts we made. So this is the paclitaxel. So this is PPF curcumin plus paclitaxel. So this is just the tumor size difference. Okay. Dr. Archana, she was a postdoc in our lab. So she also was involved in the study. You see, this is PLGA curcumin plus paclitaxel. Here PPF. The only difference is in PLGA, both are PLGAs. Here folate receptor was introduced. So when you that means the site that folic acid conjugation was done here. So this is PLGA. This is PLGA plus folic acid. So again, that size, tumor size is reduced. That means the specificity increases. Then we checked curcumin as a chemopreventive, okay, against benzopyrin-induced lung carcinogenesis. Benzopyrin is a carcinogen present in deep fried food. Okay, actually, the lung cancer is due to the main origin of lung cancer is cigarette smoke, right? So people have stopped the smoking now. Nobody smokes that much, but the lung cancer incidence is increasing. This is because of our fast food culture, because it contains lots of benzopyrin. The deep fried food, that fast food, everything has a lot of benzopyrin, which will specifically induce lung cancer. Okay, so we checked here. We cannot inject. For a chemotherapeutic studies, you can try injecting the drug. But for a chemoprevention study, chemoprevention is for a person who doesn't have cancer. Will anybody take an injection? So here we checked. It is an orally only we can give curcumin. So we gave oral dosage with the food. Okay, so we found that just giving before treating with the benzopyrin, we were giving 1 month before itself we started giving curcumin, that is as a chemopreventive. So we found that there is a very high tumor reduction in the case of curcumin treated animals. And this is Dr. Vinesh Kumar. He initiated this work. My second PhD student after Smita. Okay, he is also in US. So then we checked whether we can improve the chemopreventive efficacy using chitosan nanoparticles. Okay. So that means we compared the curcumin benzopyrin. This is nanocurcumin. That means chitosan nanoparticles. It can be this chitosan can be very safe for oral administration. So we found a very good increase in the tumor reduction occurrence. This is Janet. She is my assistant. She also has cooperated to this study. So then we checked whether curcumin can chemosensitize breast cancers. Okay, so we found that 5-FU is a drug not only used for breast cancer. It is a common drug used for breast cancer, colon cancer, etc. Okay. Why we used this selected this drug is because we found that the chemosensitizing efficacy of this curcumin and 5-FU is independent of the receptor status. Usually, when a person is diagnosed of breast cancer, the first thing asked is what is the receptor status. Okay, if it is estrogen overexpressing or HER2 receptor overexpressing or progesterone receptor overexpressing or triple negative. That means none of these are overexpressing. So if it is there is a receptor specificity to treat the cancer, it is easy. So there will be something which as an anti receptor can be given. There are so many drugs which can be given. But in the case of triple negative breast cancer, there is no specific drug to treat triple negative breast cancer. That is why we took that as a study system for our study for evaluation, because our combination did not show a receptor specificity. It was working in all the breast cancer. So we selected this system for our study. So this is triple negative breast cancer we studied, and we found that thymidylate synthase. Okay, this is actually 5-FU is working through a downregulation of thymidylate synthase. But as I told you, as a conventional chemotherapy drug, at low doses of 5-FU, 5-FU continuous use will upregulate the thymidylate synthase. Okay, that means induce chemoresistance through upregulating thymidylate synthase. So we found that curcumin, if you give 6 hours before the treatment, if you give curcumin, it will block the upregulation of thymidylate synthase. This work was in vitro combination was found by Dr. Vinod. He's an assistant professor in Kollam, SN College. And this is Dr. Harida Nair. She did the in vivo validation of this combination. This is Dr. Vinod. He was a postdoc here. He is also in US. She's going to US. So, yeah, we confirmed it using silencing the using this thymidylate synthase using shRNA and siRNA, all these techniques. So this work was recently published. So we found that curcumin can chemosensitize breast cancer cells to 5-FU chemotherapy. That also it is working independent of the receptor status. That is the most important thing you can use for triple negative breast cancer treatment. So we checked anyway, it is working in breast triple negative. So we made a carcinogenesis model. That means breast cancer is not occurring due to injection of breast cancer cells to the human, right? It is used to some environmental factors. So we made an environmental carcinogenesis model using BMBA, and we made this is an environmental breast cancer, and we tried whether. So in this case, we got breast cancers of all receptor status. Okay. So this is ER positive expressing, this is PR or expressing, this is HER2, and we got triple negative also. Okay, and we found that you see that that in the combination the tumor size is negligible. That means in some of the animals we did not get any tumor at all. So that means the combination is working in the environmental carcinogenesis model also. This work is under communication. And we found that resveratrol, a compound isolated from the skin of the black grapes. Okay, it is found in several other berries also. Okay, that colored berries also. So we found that it can sensitize. Here it was not receptor independent. We found the efficacy in HER2 overexpressing breast cancers. This effect was found in HER2 overexpressing. This is another receptor in breast cancers. This is a compound identified. The name of the compound is heteronamine. It was identified marine sponges. Okay, so this is it was found to be very effective against leukemia. Okay, this is Dr. Meenakshi Saikia. She was from Assam. Now she is in US. So along with this, our chemosensitization and chemoprevention studies, we were doing the bioprospecting. That means identifying different, we were screening different plants. I think around 20 plants we have screened for the anti-cancer potential, and we got 3 lead molecules, okay, with the translational potential. First is Chromalina odorata, that is communist pacha. This is Rhytia tinctoria, this is Dandabala, this is Solana nigram, Manatakali. So from this, it was 3 extramural projects, and this was a state-funded project. Here we got a compound, camphoride. Here also Dr. Ravi Shankar has identified this compound, and this compound was found to be very effective towards cervical cancer. And we identified another compound, tryptandrin. Here it was found to be very effective against skin cancer, both melanoma and non-melanoma skin cancer. This compound was very effective, and this compound was found to be effective. The compound utroside B was found to be very effective for liver cancer. So this is camphoride. We can see the tumor, the decrease in tumor size. Lakshmi did this work, and this is tryptandrin. So this was found from the, it was very effective against melanoma. Dr. Jayesh and Meenakshi identified this compound, and you can see the difference in tumor size. We tried 2 types of injection, okay, modes of injection, intradermal and intraperitoneal. This is the tumor in the control animal. Intradermal, that means on the site. Here we are developing the tumor. Here itself we induce the drug. Okay, give the drug. Okay, so this is the and we found that ID is a little bit more effective, but IP, that means through the stomach we are giving. Still also it is also working very nicely. Okay, so both modes of administration of drug were working very fine, very effective, the compound. So here this is to, this cell animals are not dead. Okay, we give them anesthesia, and we give, and here the cells we inject, okay, are having a luciferase conjugation. So when you inject that compound, we can see that the fluorescence. Okay, after you can take out the animal, and it will be after some 30 minutes it will be okay. So live imaging we can do. Okay, it's a imager. So the fluorescence, you see the difference in fluorescence. This is control. This is the tryptandrin treated animals. Okay, this is after opening the animal. Okay, so we can see here the, it is solid tumor. Yeah, here what we have done is, it is a tail vein model of metastasis. Metastasis means the transfer of animals, cancer cells from one organ to the other organ. So to study that, we have to here we have injected the tumor on the flank. Okay, but here we are injecting the transfected cells on the tail. Okay, but it is going and traveling everywhere in the control. But tryptandrin treatment, it has very less tumor development. It is not going anywhere that much. Okay, so we can see the intensity. This is also an animal imager, but here it is opened. Jayesh and Shabna did this work. Actually, this was a mechanistic evaluation. I don't want to give in detail. No, there is a factor called MITFM, that is master regulator of melanomagenesis. Through microarray, we found that is the molecule. Usually, all melanoma drugs are against the BRAF. BRAF, it is a oncogene, but we found that independent of the BRAF status, our compound was, it was showing a different results. It was not dependent on the BRAF status of the. So we did a microarray, and we found that it is MITFM, the regulator of the tryptandrin activity. Yeah, actually we overexpressed the animals with MITFM. It means we transfected the cells, okay, with the MITFM, and we found that then the IC50 increases. That means the IC50 was first 3 micromolar, but when we transfected, it became 15 micromolar. That means just to confirm that it is the regulator. So we have to increase the dose. But still, it was in the overexpressed cells also, the drug was working. And here also we checked whether tryptandrin can act as a chemosensitizer. That means dacarbazine is the drug currently used for melanoma chemotherapy. So see, this is the dacarbazine treated animal. You can see the combination. Actually, you may be thinking then how it is a drug. We are not using the currently used half the concentration. We are using half the concentration of dacarbazine and half the concentration of tryptandrin. So when you use tryptandrin is non-toxic, but we are getting a better effect. Here we don't find any signal also. Okay. This is live imaging. You see in the combination that MITF is completely downregulated. This is a tryptandrin as a chemopreventive against skin carcinogenesis. Okay, this is non-melanoma skin cancer induced by DMBA and PMA induced model. This is multi-stage carcinogenesis model. DMBA was used as an initiator. PMA was used as a promoter. You can see after 15 weeks here that it was full of tumors, and while the tumor number and size was very less in the tryptandrin topically applied. Look, this is Mohan. He is also in US now. So we found that it is through beta-catenin signaling. Now comes our utrocyte B. So Lakshmi did this work, and Dr. Ravi Shankar did the characterization of the work in the compound. And this was the different fractionation, active fraction, all these things. Okay, and we got a major fraction SN2. Okay, then this was given to Dr. Ravi Shankar for characterization. We got that. We found that we got a single peak. So we knew that it is a pure compound, but single spot we are getting. But we were unable to. That is another interdisciplinary collaboration. Actually, a pure chemist can do that. We cannot do that. So he did that. That is the next interdisciplinary collaboration we did. So he found that the compound was identified. It's SN2. We just gave solanum nigran fraction 2. We just gave that name. So he identified that it is utrocyte B. And we compared the activity of these the cells in different cancer cells. Okay, IC50 was calculated. IC50 means the concentration needed for 50% cell death of cancer cells. Okay, so here it was 2.5, 1.5 like that. But in HepG2, that is liver cancer, it is 500 nanomolar. Here it was micromolar. All others were. So this is HepG2. So we took this HepG2. So we screened the efficacy of the compound in different liver cancer cells. Okay, these are all liver cancer cells, while this is a normal liver. Okay, Chang liver. This is normal liver cell. So we can find that it is, we did not find much difference. There are cancer cells with HBV status, virus status, and non-virus status, but everywhere, okay, we did not find much difference. It was the IC50 was around some 450 to 600 range. So it was not dependent. Its efficacy was not dependent on the viral status. And there were up to 75 micromolar, while here it is 500 nanomolar, we are getting the IC50, but up to 75 micromolar, it was not toxic to the normal liver cells. So it is an ideal drug. One second. So you can see the tumor reduction. So this is the control animals. This is the tumor reduced tumors. The size was significantly less. So we patented this combination. I'm sorry, not the drug. Okay, it is not the drug we patented. It is the activity patent. That means the efficacy of utroside B as a therapeutic for hepatocellular carcinoma. We got a patent from US, Canada, South Korea, and Japan, and all others are pending, as he told. And we compared the efficacy. Sorafenib is a currently used FDA-approved drug against hepatocellular carcinoma. Not much drugs are available for treating hepatocellular carcinoma. So we compared the efficacy of utroside B with sorafenib. Okay, so we found that this compound is very effective than sorafenib. Okay, it is a very potent drug. So this is so utroside B, this is sorafenib. So the tumor reduction is more in our drug. Okay. In the in vitro system, it was found to be 10 times more potent, but here we did not find 10 times efficacy, but it was more potent and less toxic. And Lakshmi and Shweta did this work. So these are, this is just to evaluate the efficacy. You can see the utroside with 10 milligram per kg body weight and sorafenib 35 milligram per. It is corresponding dose of the respective IC50 doses. Okay. So it induces more apoptosis. And this is PCNA. This is a proliferative cell nuclear antigen. It will have very high expression in proliferating cells. This is controlled. This is utroside B treated. This is sorafenib treated. You can see very less PCNA in utroside B than sorafenib. Here also it is completely gone in utroside B treated. Ki67. It is a very important marker for cancer development. You can see in utroside B treated cells, Ki67 is very less. And this is another cleaved PARP. That is a marker for apoptosis. We can see it is very highly expressed in utroside B than in sorafenib. And it induces autophagy also. That means another survival signal. Actually, autophagy can lead either cooperate with the apoptosis to induce the cancer death, but it can antagonistically work also. In our study, I don't want to give that much explanation to this study. Maybe I think, yeah. We found that when you block utroside B induced autophagy, we can enhance the therapeutic effect of that means apoptosis and autophagy were antagonistically working. Okay, they were contradictingly working. That means when you block autophagy, apoptosis enhances. We use different inhibitors and methods, the biological and chemical inhibitors we use. We found that when you block utroside B induced autophagy, you can enhance the therapeutic. That means if you, now the IC50 is 500, just to know that, okay, now the IC50 is 500, but when you inhibit the autophagy, you can get the IC50 at 250 nanomolar. That means you can induce the therapeutic efficacy of utroside B if you use in combination as a chemosensitizer, any conventional, any compound which can block the autophagy. So we tried a compound, chloroquine. Chloroquine, you all know chloroquine. It is an anti-malaria drug. So it is an already used drug. So you don't have to go for any clinical trial of this compound. And we are using lesser dose. Okay, we are not in the synergistic combination. We never use the IC50 concentration. We use the subtoxic concentration so that the toxicity will be eliminated. So when we use the chloroquine in combination with utroside, we gain here it was the IC50, but here you see the combination, it is coming down. Okay, so it becomes more effective. And these are the molecular evaluations. I don't want to go into the details. You see in the combination, just to see the results of the combination. This is utroside B. This is also not the full concentration, 250 nanomolar dose we have used, but you see the tumor is completely very less, very small. Okay, so very recently, last month we published this paper, and we are now studying it in aflatoxin induced liver carcinogenesis. Aflatoxin is another, usually liver cancer was considered as a cancer for the drunkards. Okay, women and non-drinkers will not check for liver cancer. That was the condition. But now we know that there is a cancer, the liver cancer, a category of liver cancer, which is called non-alcoholic. Okay, non-alcoholic liver cancer. Okay, so that means that cancer there are different reasons for that. One is this aflatoxin. It is a poisonous carcinogen. It is found in the rotten grains, nuts, and dried food. Okay, early days, when actually in a poor family, we will in the rotten food, they will wash so many times, and what they will do, they will cook. They will think that anyway it is cooked. We all will do that. Okay, when we cook that, anyway, everything, if it is pressure cooked, everything will die, right? Even if it is a fungus, whatever will die. But the problem is that carcinogen, the toxin produced by this fungus, it cannot die. That is a chemical, right? And it is very stable at high temperature. Okay, even if you cook, it will not degrade. That means if you drink that rice water and all, if of that type of grains, and if you eat that type of nuts and all, it will only we make this model with single administration of this aflatoxin. We are not giving every day. Okay, this is a group 1 human carcinogen, very potent. This animal we are giving it's wrong. Okay, it's not 3 week. It is a 7 days old mice. Okay, so we give the start. There is only 2 doses we are giving. Okay, and it is then we are not giving anything. Okay, so but still you can see the development of, you see the big tumor developed on the aflatoxin given tumor. Okay, and this is the utroside B. We give utroside, and 6 months we are not giving, in the both. So that means it is not a chemo prevention study. It is a chemotherapy study. That means 6 months we are giving, only twice we have given, after 7 days, and I think after 1 month, 1 more times. Only 2 exposure to aflatoxin, and we keep these babies for 6 months. After that, we start giving utroside B for one group. The other group we don't give anything. But you see, with that itself, that means it can cure the aflatoxin B induced liver carcinogenesis. These are the figures. Yeah, the DNA adduct formation also, it is stopping. So this this compound, this invention has been granted, this drug, FDA granted the orphan drug for HCC drug. That means it is not still in use, but it can be used as a drug. That is that is why it is called orphan drug. It is not in the market, but it can be used as a drug if synthesized. Okay. And we have signed an MOU with the OMRF. They are very much interested in Oklahoma Medical Research Foundation for the clinical translation. We have already signed an MOU for that. And this company, Q Biomed, okay, so they bought it from us. Okay, and we got the first milestone payment. Our institute got the first milestone payment. Okay, this is the news. That's all. US FDA orphan drug designation, liver cancer. So we thought of comparing. Should I stop? Hello? Hello? എല്ലാവരും പോയോ? ആരും മിണ്ടുന്നില്ല. We compared the efficacy of the compound, actually we have already sold out the compound, right? It is already given. But I was feeling, if you don't give also, nobody is going to work on that. Okay. In our India.
Sorry,
Hello?
ma'am, for the interruption.
Yeah.
You can continue.
Okay, thank you. Thank you. Okay. So actually, this compound is already given to a multinational company, so they can only sell it. But first I was thinking, okay, we will do that clinical trial. But to do a clinical trial, millions of money is needed, and I am going to retire next year. So it's not possible for me to make it into a drug by myself. Okay, and lot of money, and more than money, it is the rules and regulations pertaining in our country. It's very difficult. So anyway, we transferred. At least they will do it. But in my conscience, I have a feeling that for our people, if we can, this utroside B, not like curcumin or the other compound, it is completely soluble in water. Utroside B is completely soluble in water. So I thought, okay, if it is soluble in water, this leaf, we are getting this utroside B from the Solana nigrans leaves, right? So if you give the amount needed to isolate this 500 nanomolar utroside B, it should be there in the leaf, right? So we are giving we started a trial. It is a toxicity evaluation. Actually, we cannot get a permission for a clinical trial, so we have taken a toxicity evaluation. That means people who actually this drug is being given, this not drug, this leaf is given as a medicine for liver related diseases by naturopathy and Ayurveda doctors, and they we collaborated with them. So we are taking we are analyzing the blood, the liver function, kidney function, and lipid profile of the patients involved in their study. So this is a collaborative work with one Ayurveda and naturopathy doctor. So people who are interested in, after seeing our results, people are approaching us for our, asking for utroside B. We cannot give utroside B, definitely. So they are joining in this study, and we are comparing their evaluate, and it is a toxicity study, so there should be healthy volunteers also. So in this study there are healthy volunteers, people with the liver related diseases like fatty liver, all these things, then people with the chronic liver disease, okay, that means NASH, and the other is people with the liver cancer. All people are there. Okay, so we did not find, and we are checking their kidney function also to make sure that it is not toxic. And there are, it is a saponin. Most of the saponins are shown to be having a hemolysis effect. We checked utroside B, whether utroside B and sorafenib is doing a hemolysis. That means lyse the blood cells, but it is not doing. This is the positive control. We did not find any hemolysis with both these compounds. Here this work is done by Shweta and Regina. Here this is patients. I mean, you can see so many patients are there. None of them got a hike in their liver function, kidney function, or lipid profile. Okay, that means the extract is completely safe. Okay, that means the leaf powder containing IC50 concentration of utroside B. This is their lipid profile. We are looking at very high reduction in their lipid profile, cholesterol, all these things. Okay. And this is AFP and PIVK. This is tumor. This AFP and PIVK are the markers for liver cancer. This is 1 month uptake. Okay, so all the people have a reduction in their AFP levels. His is completely going down within this month. In the 60 days, it has come here. Okay, so it is reducing the tumor markers of liver cancer markers. Yeah, this is a NASH model. Regina is doing that. It is currently going on in our lab. You can see that the NASH, the treatment reduces they are given a NASH model. We are giving high fat diet. Actually, fatty liver is produced by fatty food, right? So that means we are giving fatty food to these animals. This is NASH animal. So you can see the high fat accumulation this is extract given, we don't find here. Yeah, here also we can see the difference. Here this is the collagen accumulation in the high fat diet. This is the liver of the animals. You can see it is gone. This is actually, I'm not supposed to show this slide because the patient data, we are not supposed to expose to anybody. But this is of a person from our institute. He gave his photo also to put in this to get it more. He want people to see that he is cured because he was taking only our medicine, nothing else. Okay. So he was diagnosed of chronic liver disease. That means cirrhosis. Okay, almost. So in 2019 he started taking our capsule. That means leaf powder. This is it reduced. It was reversing to grade 2 fatty liver. 2020. Now he's grade 1 fatty liver. Okay, so that means he is really cured. So he's so happy that he gave us the report and gave his photo also. Anyway, I removed that. It is unethical. So this is actually this person, this Niranjan. His name was Niranjan. Actually, he started this study actually. When after seeing our report in our publication in scientific report, it is a Nature journal. He came from Pune. He is from Pune. He came to our lab from Pune only for that, and he asked us whether I can get the utroside B because he has been asked to do the liver transplantation and he was not getting a donor. So he told, Madam, I have to live till I get a donor. So please make me. And he was taking sorafenib and he was telling, I am resistant to this drug and I am feeling very tired. Actually, sorafenib is very toxic. Even the animals in our sorafenib group was also, even though they are showing tumor reduction, they were showing the blood stains in their fecus. They were showing very tired, found to be very tired while our utroside B people were running around. Okay. So he told, I cannot take sorafenib anymore. So can you make me alive till I do my transplantation? So we told we don't have a trial also. We have just started our animal experiment. He told, anyway, I have no other option. Please give me the leaf powder. And so we gave him. So he sent I told him to check it after 1 month, but even in 15 days itself, he checked. So he's telling his AFP. It is a marker for liver cancer. I told you already. It came from 60 to 45. Then it again, it was 30. Then up to 15 it came. But after his transplantation, we don't have any contact with him. We don't know what happened. So anyway, it was really, it gave us a motivation to do this study, this trial. So this is Dr. Riel Turner from Oklahoma Medical Research Foundation, who is collaborating us for the clinical validation of this Utrocyte B. And this is my lab. Okay, so I think I introduced almost everybody. These were all my PhD students. Now they have took their PhD, and Shabna is continuing as a research associate in my lab currently. And these were my postdocs who worked with my lab. Now he has joined as a MK Bhan fellow, and this is my current lab. Now only this many people are there. Shweta has almost completed. Aishwarya, she's a it is a collaborative project. Not my PhD student, I am her co-guide. So Dr. Smida's student, and this Kirtana and Regina are currently working on Utrocyte B. Thank you.
Thank you, ma'am, for this inspirational talk. I request Dr. Anto PV to take over the session.
Floor is open for. Thank you, ma'am. The floor is open for the queries. All the participants can make their questions now. This time.
So we have a question. It's from Manu. The question goes like this. He have two questions. While using animals as specimens, what are the legal formalities that are need to be addressed? That's the first question.
Okay.
And the
Can I
second question.
answer that? Can
Yes,
I
ma'am,
answer
yes,
that?
yes,
Yeah.
yes.
Actually, there is an institutional ethical committee. In every institute, it will be there. Okay. So this committee will have representatives from the institute and institute representatives from our all other committees of this animal welfare. All this. There will be a government nominee. So many people will be there. Okay. So we have to present the work in this committee. We have to explain why we are doing this experiment, and there are some guidelines called OECD guidelines. So we have to plan our experiments according to OECD guidelines. So that is the protocol.
Okay,
Yes.
the second question is what are the challenges in such an age of animal rights as well?
I did not get. What are the challenges?
In such an age of animal rights.
Age.
Yes.
Yeah, that's what I'm telling. There will be that animal rights committee representative is one of the person here. Okay. There will be, yeah, actually they will ask us, anyway, you are taking 6 animals, control animals, why you are taking, always the control animals will not get cancer, why you have to take all these things they will ask. But if you don't have a control, how can you tell that this drug is effective? That means you have to repeat the whole study, right? So that all these things we have to explain. Okay. If you for every time, they may ask, anyway, the last time you got tumor, this time also you will get, then you treat only keep only the drug treated group. That's also impossible. There can be individual variation, there can be time variation. That's some animals there will be there is a phenomenon called leaking. That means these immunocompromised mice, after some time, they get the leaking because their immune deficiency loses. That means the tumor will not grow. So if you are giving only the treated group, if the tumor is not developing, we will be thinking that our drug is active. But actually that is not the case. The animal is gone. Okay. So each and every time we should have proper controlling. Otherwise you cannot get a good result. You cannot publish in a good journal.
Okay, ma'am. Another question is there. That is also from the same person. He is pursuing a research scholar in studies in medical humanities. He
Okay.
would like to know how much doctors of today could treat patients, particularly cancer patients, as subjects rather than specimens.
That right. I cannot tell that answer, right? I'm not the person to answer that question. I am not a doctor. Okay. I am a scientist. Actually, I for these type of studies, I am collaborating with doctors. Definitely for the breast cancer work, I am collaborating with a doctor in Dr. Srikumar Pillai from Jubilee Mission Cancer Medical College, Thrissur. I think it is a very productive collaboration because he is giving us the breast tissues removed. It's not a normal breast. It is not breast cancer tissues and liver cancer tissues. So because they are taking that pain, most of the cancer doctors will not do that. Okay, why to waste their precious time? But he is so good that he before the surgery he informed us, and my students travel from here, and we book all the the travel all these things, and we take a liquid nitrogen container, we bring the whole thing in an organ transplantation medium, and here we have a veterinary surgeon. He is so dedicated that when this student reaches here, I will go and get them from the railway station. It is 9:30. Okay. Then we do the surgery here to the animals, immunocompromised animals, and I do the primary isolation. And when I go home, it is around 3, 4 a.m. in the morning. So it is all dedication. Okay. If we don't do that, also nobody will tell anything. But it is, we feel that it has to be done by somebody has to do that, right? Then only otherwise always it will be in animals only. It should be transferred to human, right? We are doing this work only not for animals, right? The animals are sacrificing their lives to for human beings. Okay, when you kill an animal, you don't tell that you kill the animal, you sacrifice the animal. That is the term used. Okay, so it is for the benefit of, in all the communities, all the orders, whatever profession, there is good people and bad people. Those who just work for money, those who dedicate their lives to for that job. So it is there always. So it is your approach, what to do, why to do, how to do.
Okay, ma'am. Thank you
Yeah.
so much for answering the questions.
Okay.
I request Anto sir to take over.
Thank you, ma'am.
Okay.
I had one matter from the relation with the St. Thomas Institute with you. That he is
Yeah.
our former
Yeah, yeah,
former
yeah, yeah.
faculty member.
I have a very big, not small relation, very, very big relation. Yeah.
Okay,
മരുമോളാണ്
ma'am.
മരുമോള് കേട്ടോ അവിടുത്തെ.
Okay, okay.
ഏ?
I have also a good experience that I have a chance to teach your
Ah.
elder son,
Oh,
Nicolando, in plus
oh,
two.
yeah.
Okay,
What's
ma'am.
your name, sir?
Endo
Your
PV.
name?
Endo
Oh,
PV.
okay, okay, okay. Yeah, yeah. Okay, okay, okay. Because I don't see, I am seeing my screen only. I don't see the face, anybody's.
Okay,
Okay.
okay, okay, ma'am.
Yeah, that is, can I, 1 second. It'll stop you. Okay, 1 second. Yeah,
Yeah, yeah.
actually I don't see anybody. Yeah, who is you are Anto sir?
Yeah, yeah, yeah.
Yeah, okay, okay, okay. I will tell him.
Okay,
Okay,
okay, okay, okay.
okay. I don't see anybody. Where is Anne Mary?
And Mary, come, come here.
Uh,
Where you?
Yes, ma'am.
remove your mask for 1 second. Mask, mask. Yeah. Hello. Okay, okay. I just wanted to see. That's all. Okay. Thank you. I cannot hear. You are not audible. Yeah. Okay. Okay. Thank you. I cannot hear. Yeah.
Yeah.
Yeah, thank you, ma'am.
Okay. Thank
We
you.
can wind up the program.
Yeah, okay.
I am sure that all of the participants are very much inspired by your excellent presentation,
Actually, how
especially
many
in
people
the
were there? Can I
nanoparticle.
know? How many people were there attending?
How many people participated? 45. 45.
Oh, okay.
All are research fellows.
Okay.
150 research fellows here. Then out of 50, only the science research fellows are participating in this program.
Okay.
Okay, madam. I thank you on behalf of St. Louis College for the valuable time and effort you have spent for this session. Thank you, Ruby ma'am. Thank you once again.
Thank
Thank you,
you.
ma'am.
Thank you, Dr. Anto PV and Dr. Ruby John Anto. By this, we come to the end of the day one of Research Scholars Meet and National Seminar. Tomorrow, we'll begin the session at 9 a.m. and we'll meet tomorrow. Thank you.